Hello friends, myself Dr. Brijesh Mishra. Today we are going to talk about the novel coronavirus which has created a pandemic situation and currently we are in this crisis. So I will discuss about its epidemiology, pathogenesis and the possible ways to combat this disease in this class. So first of all we need to know about this pathogen, its origin, mode of action, etc. This virus belongs to the family of Coronaviridae, in which other members are severe acute respiratory syndrome coronavirus and Middle East respiratory syndrome coronavirus. The WHO has given it a name that is SARS coronavirus 2 because of its similar action as that of SARS coronavirus. And the disease caused by it is called Covid-19. Since the outbreak has started in the year 2019, so the name is given Covid-19. The first cluster of cases of Covid-19 was reported in people associated with wet meat market in Wuhan, China, where live or freshly slaughtered animals were sold. The first host animal through which it enters human is most probably a bat. Why bat? Because the genome sequence of bat coronavirus is about 95% similar to that of SARS coronavirus 2 genome, and the intermediate host between bat and human being is most probably a Himalayan civet cat. As we have seen that its pathogenic action is similar to that of SARS coronavirus, its structure is also somewhat similar to SARS coronavirus. As you can see that the pathogen comprises spike proteins all over its membrane, which play a critical role in binding with the host cell. Also there is envelope protein and membrane protein which forms the capsid for this virus, and the genetic material of this virus is single-stranded positive RNA, which is around 3000 base pair. There are two types of spike protein, S1 and S2. S1 binds with ACE2 receptor while S2 mediates fusion between virus and host cell. So till now we have seen the origin, the structure of this pathogen. Now understanding the pathogenesis is very important. See, when it enters human lung, the spike protein 1, that is S1 protein, binds to the ACE2 receptor of type 2 cells of the alveoli. And at the same time, the S2 protein helps the pathogen to get fused into host cell. As you can see in the diagram, once the virus enters the host cell, the protein parts get broken and the RNA is released into the cytoplasm of the host cell. Now this RNA utilizes translatory mechanism of the host cell to make its protein, that is envelope protein, membrane protein and spike protein, so that it can be assembled into the capsid form later on. And parallelly this RNA uses RNA dependent RNA polymerase to make its multiple copies. And once the multiple copies of RNA and proteins are made, they all get assembled into its original pathogenic form or in a vesicles, and they get released from the cell through the process of exocytosis and start infecting the other cells. This all process results into the accumulation of dead cells, proteins in the alveoli of the lung, and which results in inflammation, and it causes acute respiratory disease. COVID-19 is one of the most contagious disease of the human race. Why I am saying this? Because its incidence rate is so high that in the current scenario, it is affecting around 1 lakh people per day in the world. And again, the question arises why it is so contagious. Because it's high sustainability on the non-living things also, that is for more than 72 hours, which makes it more contagious. Currently, there are more than 14 lakh cases reported all over the world, and out of which 70,000 deaths is also been reported. The fatality rate is around 5.5%. That's why on March 11, WHO has declared it as a pandemic situation. And the most affected part of the world in current scenario is USA and European provinces. Only USA has reported around more than 4 lakh cases, and in the Europe, Italy has reported the highest number of death, that is more than 17,000. The cases are increasing day by day, and there is no sign of a stop we can see right now. And some studies have also projected that there can be a huge number of cases which can create a serious issue. The major problem is still we don't know much about this disease. We know that the disease can be transferred through mucus or fecal medium, or one can get it if he touches a affected person, or a affected person sneezes and other person gets to them in a close proximity. These are the chances that the disease can be spread. And we also know that the pathogen can survive for more than 3 hours in the air also, and for uh, more than 72 hours on other surfaces. We know this much, we know, but still there are so many things we don't know. Like we don't know, can a same person can get affected for more than one time, and after recovery can the virus regrow in our system. So these are the things, and due to its high sustainability, it seems very challenging to eradicate it from our environment. Another problem is that most of the time there is no symptoms in the initial period, due to which a person can unintentionally spread it to others. The common symptoms which are seen in these COVID patients are fever, cough, fatigue, and short of breath. Once the symptoms appears, then only one prefers to go for testing. But the better way will be to test a large population to detect the real problem or real picture of this pandemic situation. The testing is done by collecting nasal swab, isolating RNA, making cDNA, and then performing real-time PCR to detect the genome of virus. Looking at the treatment part, this is very unfortunate that there is no as such specific drug or vaccine which has been approved worldwide to treat this disease. However, several agencies have claimed that they are very close to make such drugs or vaccine which can be useful to treat the COVID-19. Meanwhile, in several parts of the world, hit and trial methods are going on to treat COVID-19 patients. The common drugs which are used are hydroxychloroquine, lopinavir, ritonavir, etc. Hydroxychloroquine can be useful as it can increase the endosomal pH, which results in inappropriate binding of the virus and host cell. That means the coronavirus and the type 2 cells of alveoli cannot bind, and it will prevent the growth of the virus inside the host. The drugs which are used to treat HIV patients like lopinavir and ritonavir are seen to be effective in COVID-19 patients. Also, it has been seen that plasma from recovered COVID-19 patients can be very effective in treating the critically ill COVID-19 patients. And FDA has approved it for only critically ill patients. There are several steps in the pathogenesis of this virus which can be targeted to make a potential drug for this disease. The first one is that spike protein binding with ACE2 receptor can be inhibited by enforcing competitive binding by injecting complementary antibody with respect to ACE2 receptor. Similarly, RNA-dependent RNA polymerase can be blocked so that replication of virus genome can be inhibited. Also, antisense RNA technology can be used to block a particular fragment of the virus so that it cannot form all the necessary proteins required to form its capsid. It has been very well said that prevention is better than cure. In case of COVID-19, we can fight with the disease by preventing us from the exposure of this deadly coronavirus. For that, social distancing is a must, proper hygiene should be maintained, proper cleaning should be there, and the awareness should be spread so that whole community can come together to follow these steps. So guys, this was the class regarding COVID-19. Please let me know if you have any doubts. I will be back with the updates on the disease. Till then, take care.